where is it born? Where was it born in this
Thymus.
okay.
Primary.
It originates in the bone marrow but developed mature in the thymus. What will happen with the mature T cells? What we call naive T cells. It's finished the maturation, but still in the thymus. What will be the next step? Somebody told that it will get to the circulation. And what will be the goal of this T cell? Go to the secondary lymph nodes. Okay, why? Why exactly to the secondary lymphatic
To meet the
organs?
antigen.
To find a specific antigen. Yeah? How many T cells develop per day? Do you know it? In the thymus. Billions. It's not a bad response. Any other? Millions. It's not a bad response. So we don't know exactly. That's what I want to tell you. Okay. 10 billions to 100 billions. A lot. That's the answer. One B cell. How many antigen can be recognized by one T cell? Sorry. One T cell can recognize only one antigen. How many T cell receptors are on one T cell? A lot again. About 100,000. It's a more exact number. But these 100,000 receptors can recognize the same antigen. Okay, so one lymphocyte for one antigen. Okay. But the 100 billions T cell, how many antigen can be recognized by the 100 millions? 100 billions. Okay. So one T cell alone cannot protect the human body, but the 100 millions will recognize everything. Yeah? So all the lymphocytes ask just one question. Okay. Is it a listeria? No. Then the next lymphocyte comes. Is it a listeria? No. Then the next one. One of them will recognize it. Okay. And where can a T cell find an antigen? A naive T cell. Only in the secondary lymphatic organs. Okay. A naive T cell will not look for the antigen in the tissue, in the skin, in the intestinal tract, only in the secondary lymphatic organs. How can it get to the secondary lymphatic organs? To the circulation, but how? I'm just swimming in the blood. How do I know that that's a secondary lymphatic organ? We are lymph nodes, okay. Homing. What does it mean, homing? So in the lymph nodes there are a special endothel. What we call high endothelial venules. And in these venules there are special anchoring molecules which grab the lymphocyte, the naive lymphocytes. Okay? So naive lymphocytes cannot exit anywhere, can exit only in the secondary lymphatic organs, in the lymph nodes. Okay? What will do the T cell in the lymph nodes? No, it matured in the thymus. What will do in the lymph nodes? Look for the specific antigen. If I could find my antigen, the only one, then the T cell will activate. What will happen with the non-specific T cells? Continue look for the antigen. Where will it go? To the next lymph node, to the next lymph node, by the lymphatic vessels. After it, what's the end of the lymphatic vessels?
It's a dead end.
Okay, the beginning is a dead end, and what about the other?
The vein.
The vein, okay, subclavian vein, okay. So go back to the circulation. From the circulation, exit again another lymph node. Check the lymph node. Can I find my antigen? No, you know. Go to the next one, next one, next one. What will be the end of this process? Apoptosis. In a few weeks, the non-specific T cells will die. Every day, 100 billions of T cells are produced in the thymus. Every day, 100 billion T cells die in the periphery. Okay? We don't have more and more T cells for sure. So every day, the T cells die. Okay, so non-specific T cell die, specific T cell start to proliferate. What's the name of this proliferation? It has a special name. What's the reason of this proliferation? So we have one specific T cell. Cloning. What does it mean, cloning? Clonal proliferation, that's the name. Clonal expansion. What's the reason of it? Why is it good that it's clonal?
They're having more number of same cells.
Have the same specificity. Okay? So we have one specific T cell at the beginning of the response, and we have 1 million specific T cells with the same specificity in 1 week in 10 days. So why is it that the adaptive immunity is so slow? It requires a lot of time because of this proliferation. We have at the beginning of the response we have only one specific T cell. It requires 10 days to produce cells enough. So that was the life of a T cell. It's okay. What a T cell can recognize? What kind of molecules? What will be the antigen for a T cell? What is recognized by a T cell?
MHC2 complex.
Okay, so good. MHC molecule. MHC 1 or 2?
2.
One or two, it depends. Okay.
Well, if it has nucleus, it's MHC1.
What kind of cell express MHC1?
All
All the nucleated cells. What kind of cell express MHC2? Professional antigen presenting cells. Can you list me?
Macrophage.
Macrophage.
Dendritic cells. No.
B cells.
3 professional antigen presenting cell. Dendritic cell, macrophage, B cell. We will come back. So focus to the intracellular pathogen, intracellular infection. How can a cell detect that it's infected? Any somatic cell can detect that the cell was infected. How is it possible?
Continuous expression.
Okay, by MHC1, any other way?
Lack of MHC.
That's good, very good. Pumps. What does it mean, pumps? If a virus infects a cell, the double-stranded RNA, viral RNA, and so on. So all the human cells express pattern recognition receptors for intracellular pathogens. Okay? For extracellular, no, but all the cells can recognize intracellular pathogens. What will be the response if I recognize that I am infected?
Apoptosis.
Induce apoptosis, induce antiviral mechanism, so block the viral replication, and you reduce the RNA synthesis, reduce the protein synthesis, induce RNA degradation. So any cell can recognize the intracellular pathogen, will produce type 1 interferon, and you remember that? And type 1 interferon will block the viral replication, induce apoptosis, or ask a kill me signal, activate the cytotoxic T cell, the natural killer cell. Okay, so we have a system where all the nucleated cell can all the human cell can recognize infection, but it's a somatic cell. It's not a professional cell. Yeah? So the goal of the antigen presentation that the immune system, the professional cells, wants to see what is inside the cell. The immune system can see the extracellular pathogen, very easy, yeah, just to see by pattern recognition receptor, easy to see the extracellular pathogen. How can detect the immune system what is inside?
Expression of peptide fragments.
Expression. So one theoretic possibility that the immune system gets into the cell and look what is inside, but it's too difficult for sure. So that's why all the intracellular molecules are presented on the cell surface. Okay? That's the theory. After the evolution started to say that do I have to see all the molecules? Is it important to see everything? Lipids, DNA. You can take that table.
The what?
That table. Let's see
Yeah,
on that.
yeah.
Or any other table.
It's cool. Do we have, is there chairs?
Yeah, like a chair.
We can just sit on the table.
Yeah, sit on the table. That's what I Okay, so we have a virus or bacteria or anything in the cell. Is it important to put on the cell surface all the virus?
Just part of
Or a part of it? What part of it? Only proteins. Is it important to express all the proteins?
Just for
Only peptides. Okay, so that was step by step the evolution. What will happen with a free peptide in a soluble peptide? If we put a peptide in water, what will happen with the peptides?
Bind to.
How will the conformation of the peptide change with 3 billions of conformation? Yeah, peptide can turn any direction. So that's why to recognize a peptide, it requires a chaperone molecule. That's the MHC. Okay? So the MHC grabs the peptide and fixes the conformation. That's the only role. So that's why all the nucleated cells express MHC1, grab the peptide in the intracellular area and present it on the cell surface. Okay? So all the nucleated cell can present what is inside. Antigen presentation depends on the infection. Depends whether the cell is infected or not.
Yes. It does.
No.
No.
They always.
The goal of the process was to present for the immune system what is inside. Okay? It's not the decision of the cell that I am infected, I just want to present. Write on my t-shirt that that's in me. Okay? And the T cell will decide that is dangerous, non-dangerous, pathogen, non-pathogen. Okay? So antigen presentation is going all the time with the same intensity, more or less. Okay? So we have a virus in the cell. How can we create peptides from the virus?
Breaking down the proteins,
The RNA.
No. So we have virus, we want to create peptides. From the proteins, how can we do peptides? We have a special organelles. The cell has special organelles.
lysosomes.
All those lysosome, the name is proteasome. Yes, proteasome. Okay. So the proteasome can distinguish pathogen, non-pathogen, human, non-human. No, it's a stupid enzyme. Just cut the proteins to peptides. Okay. So proteasome will cut all the proteins what is inside the cell. Okay. Any protein has a half-life. Sooner or later, the proteins will be cleaved by the proteasome. So we have peptides in the cytosol, and we have all the proteins are synthesized in the ER. So we have MHC1 molecule in the ER, we have peptides in the cytosol. How can we do a love story to find each other?
Transport.
Transport system. The name of this transport system is TAP, the peptide transport. These TAP proteins are the membrane of the ER and just transport proteins from the cytosol to the ER. In the ER, the MHC1 molecule grab the peptide and bring it to the cell surface. What will happen with an empty MHC1 molecule without peptide? In the ER, there are a lot of MHC1 synthesized all the time. What will happen with it? It's very logic, cannot go out, okay? Only by peptide. When the MHC1 grabs the peptide, the conformation of the MHC1 is changed, drastically changed, and this MHC1 with peptide will be transported. On the cell surface, can we change the peptide? No, it's absolutely illogical, okay? Because if we could change, then it would be an extracellular peptide. So the MHC absolutely fixes the peptide. It's a very strong binding. Okay. So to summarize the intracellular presentation, all the nucleated cells express MHC1. All the nucleated cells can present what is in the cell. All the proteins in the cell will be cleaved by the proteasome. The created peptides will be transported to the ER, and the MHC1 presented for the T cells. So what kind of T cells can recognize the MHC1 peptide complex? The cytotoxic T cell, and what do you do with the cytotoxic T cell with the antigen presenting cell? Induced cell death, so killing. Yeah. If an antigen presenting cell present without peptide, it means that the cell was infected, must be killed. Okay, so cytotoxic T cell kill all the nucleated cell can be killed by the cytotoxic T cell. What about the non-nucleated? What about the red blood cells? Why spleen? That's good, okay. So in the spleen the macrophages will eat it, that's good. But why these cells do not require MHC1? Can a virus infect the red blood cell? In theory can, but what will happen with the virus in the red blood cells? There is no protein synthesis, nothing happen. Yeah? So virus cannot replicate in the red blood cells. So that's why the red blood cells are not in danger. They do not require MHC1. Okay, so that's about intracellular protein. How shall we present extracellular proteins for the T cells? MHC2 molecule. Is it important that all the cells express the extracellular area, the extracellular antigens? All the nucleated cells can present intracellular, all the cells can present extracellular.
No.
No, for sure. What kind of cell can present? Macrophages, dendritic cells, and B cells. Currently focused to macrophages and dendritic cells. These cells can do a lot of things which is important for extracellular presentation. First, these cells are phagocytic cells. So all the time check what is outside. A fibroblast can take something from the extracellular, or a hepatocyte, or anything, or a neuron. Normally not, or not too much. So how could a neuron present extracellular antigen? It would be very hard to solve. Okay? So that's why macrophages, dendritic cells were present. Macrophages and dendritic cells are very good in recognition. So if there is a pathogen, these two cells will phagocyte it. A neuron will not phagocyte a pathogen because don't have extracellular pattern recognition receptor. Okay. And where can we find in the human body macrophages and dendritic cells? Everywhere, all the tissues. So anywhere we have an extracellular pathogen, these two cells can present it. Okay? So that's why these are the professional antigen presenting. Macrophages, dendritic cells, and T cells. Next week we will see the B cells. Okay. So the first step of the extracellular antigen presentation, that macrophages, dendritic cells recognize the pathogen and engulf it. Yeah? How can we create peptides from extracellular pathogens?
Same process.
So when you phagocyted something, these molecules get into the endosome, phagosome, and it will fuse with the lysosome. Okay? So the phagolysosome, what kind of enzyme can you find in the phagolysosome? Peptide? Protease. Yeah. Protease, that's good. So it's full with protease. All the proteins will be cut to peptides. In the phagolysosome. Where are the MHC2 at this moment? In the ER. So that's a problem. How can the MHC2 find the peptide, which is in the phagolysosome? So there are two possibilities. The peptide must go to the ER, or the MHC2 must go to the phagolysosome. The MHC2 will go to the phagolysosome. Okay? Good. So MHC2 can bind peptides, only the phagolysosome transported to the cell surface. There is a big problem. How can we solve that the ER is full with peptides, because the top system transport all the time from the cytosol, but the MHC2 mustn't bind intracellular peptide, only extracellular. Yeah? MHC2 is for extracellular pathogen. So, just by chance, any one of you know the structure of the MHC1? How does it look like? Very good. MHC1 has an alpha chain and has a beta-2 microglobulin. The beta chain has no any role, it's just for structure. Okay, the alpha chain alone can bind the peptide. So in case of MHC1, one protein, one alpha chain bind the peptide. How is it in case of MHC2? We have alpha and beta chain. two different proteins. Alpha chain alone cannot bind the peptide, beta chain alone cannot bind the peptide. Okay? So in the ER we have alpha chain, lonely alpha chain, lonely beta chain. The ER is full with peptide, but alpha chain alone cannot bind. Is it okay? How can the two chains find each other, the alpha and the beta chain? With a chaperone, that's what we call invariant chain. With the invariant chain, attach the two proteins to each other, and in the same time, the invariant chain occupy the peptide binding group. Okay? So in the ER, we have alpha chain, beta chain, lonely, or we have full MHC2, but we block peptide binding group. Is it okay? So these complex, the MHC2 with the invariant chain, are transported to the phagolysosome. What will happen in the phagolysosome? What will be the first step? We have a block MHC2. It must open. Why will it open? It will not open. There are a lot of protease. Yeah? And the protease will clip the invariant chain. Okay? And that's why the peptide binding group became free, can bind peptides. Okay? So, because MHC2 has two chains for peptide binding, it cannot bind peptides in the ER, it can bind only in the phagolysosome. Okay?
Invariant chains and the variant chains.
Invariant chain, not variable. All the MHC2 bind the same chakra. So, can you guess how many MHC molecules are on the cell surface? Millions, not billions, millions. How many peptides can be presented in the same time? No? 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 Millions. Millions. Okay. So the MHC molecule can present one peptide. We have millions of MHC molecules can present million peptides. These peptides will be the same on the surface of one cell. We have million MHC molecule present, all the molecules present the same peptide. No, it's absolutely random process. The cell is full with peptides, some of them will be presented. What will happen if a cell contains very few virus, one virus? How many peptides will be presented out of the virus? How many presented out of the human? It's a very weak infection. Nothing, or one peptide out of the million will be a viral peptide. What happens if the virus is replicated in the cell? We have full with virus. A lot of viral peptide will be presented. What if the cells are not infected? Only human peptides will be present. Antigen presentation is an automatic process all the time going. If the cell is infected, then viral peptides are presented, and human peptides, not then not. Okay. So that was the extracellular antigen presentation. We should speak about the structure of the T cell receptor.
MHC, if you will ask.
We finished the MHC.
No, receptor.
So that MHC peptide complex is recognized by the T cell receptor. Okay. What is their structure? Do you know the structure of the T cell receptor? Alpha chain and beta chain. And what is the domain structure? Okay, that's the BCR, B cell receptor, what we talked about. T cell receptor don't have light chain, heavy chain, has alpha chain, beta chain. Okay? Alpha chain has one constant domain, one variable domain. Beta chain has one constant domain, one variable domain. And the two chains are covalently attached to each other by cysteine bridge, by this sulfate bridge. How many variable domains are in the human body? Billions. We have billions of T cells. Each T cell recognizes different antigen. Each T cell expresses different variable domains. Okay? And how many constant domains? The constant is constant. So we have only one constant domain. The variable, as the name indicates, is different in all the T cell. Constant domain is the same in all the T cell. Okay.
These are the questions on the exam. These questions you're asking is going to be on the exam?
Yes, I will see. I tell you only what I can ask. Hmm?
Like millions, billions. Be more specific.
Not exactly millions, billions, but variable must be a lot. Yeah, most of it. You must know the structure. I can ask the structure. T cell receptor has one alpha chain, one beta chain. You must know. B cell receptor has two heavy chain, two light chain. The B cell receptor. Okay. So don't mix the B with T. It's crucial. Okay. That's an absolutely possible question. List me receptors which has two chains. Only two chains. Tell me receptors with two chains. B cell receptor has four chains.
No, MHC2.
MHC2. T cell receptor. MHC1. Yeah. So that's such kind of question.
MHC1, you said it's like one.
MHC1 has two chains, but only one bind the peptide. That's another possible question. Tell me receptors which bind the peptide with two chains. Not that easy, not that easy, yes, of course. I won't ask only what kind of cells are the phagocytic cells.
What was the last question, sir? The receptor bound peptide is only
Two chains.
Two chains. Okay. So the T cell receptor alpha and beta chain cannot bind the peptide only together. Okay, alpha chain and beta chain together bind the peptide. That's the MHC peptide complex. Good. We finished the intracellular antigen presentation, extracellular antigen presentation, and T cell receptor structure. So we are quite good at this moment. Tell me the function of the T cells. So what's the function of the cytotoxic T cell? That's quite simple. Kill. To kill the antigen presenting cell. What will be the mechanism of it? I will need two different processes.
Apoptosis
Okay, so induce apoptosis by fast ligand system, by death receptors. Okay, so by cell contact, the fast ligand can kill the
Four
fast.
capsules, 3, 10.
Yeah, you don't have to know. Just by death receptors. What kind of cell express fast ligand in the human body? Only the killer. Who are the killers? T cells and natural killers. Nobody else express fast ligand. Which cell will express fast? The receptor? All the cells. To, if you want, kill me.
Can you repeat?
Suppose the T cell and the NK cell kill the target cell by fast ligand.
Do we need to know the structure of the receptors?
No. What I tell you, you know, must know. What I don't tell you, don't have to.
So sir, the ligand is not T cell and natural killer cell, and the receptor is not.
Practically more the same. Okay. So till this moment you said me one mechanism. Induce apoptosis by death receptors. How else can the T cell kill? Perforin granzyme system, so the T cell has vesicles, preformed vesicles, and release it, the perforin and granzyme. The perforin is a pore-forming protein, just do pore on the cell surface, and granzyme can get into the cell. And granzyme and proteases just leave the cell structure. Okay?
They do it in the mitochondria or in the cell? Because they make it pour on the mitochondria.
No, on the cell. Don't mix it. That's a process of apoptosis. That's completely different. The T cell wants to kill the cell, not the mitochondria.
Bad.
BCL2 don't mix it. We are not here for apoptosis, we want to kill the cell. Okay, so the T cell can kill the cell by direct cell-cell interaction using fast ligand system or by releasing vesicles using perforin enzyme system. Okay, that's for cytotoxic T cell. What will be the role of the helper T cells?
Mediated.
Help B cells, what else? I couldn't hear you.
Mediating cytotoxins.
Regulation.
Activating. A lot of
Okay.
things.
Several jobs.
All the response was good, altogether is still not enough.
What do you want to hear?
So the first, helper T cell, not to kill the cell, but to help, will produce cytokine. Okay? Cytokine to help for the antigen presenting cell, mostly the antigen presenting cell. Okay? We share the pathogens to different groups. What kind of cytokines would be optimal against parasites?
Or that's for TH2, cytotoxin.
So what kind of cells are active against parasites?
Mast.
Mast cells, eosinophil, basophil, granulocyte. So if a body recognizes a parasite, helper T cell will produce IL-4 to activate the basophil, eosinophil mast cells. Okay? That's what we call TH2. It's interleukin 4.
TH2.
Interleukin. So we have different kind of helper T cells. Yeah. One group of helper T cell will assist to the anti-parasitic response. That's what we call TH2. Yeah. Another group will assist against intracellular pathogens. That we call TH1. And the third group will assist against extracellular, which is TH70. Okay. Shh, can you be quiet, really, or I don't know. Turn to the wall, or I don't know. As far as I can see, some people would like to listen to me, so let them to work. Okay. So, at the beginning, I shared the pathogens into three groups: intracellular, extracellular, and intercellular bacteria or parasites. Okay, and we have three kinds of helper T cells. And once more, okay, so against parasites, TH2 will produce IL-4, interleukin 4, to activate eosinophil, basophil mast cells. Okay, that's everything is on the slides. Okay. Finished? Okay. So against intracellular pathogens, TH1 cell produce interferon gamma and will activate, what will activate the interferon gamma?
Macrophages.
Okay, so you have two ways. At first memorize. Okay. The second, we want to kill intracellular pathogen. Who can kill the intracellulars? Cytotoxic T cell and T cell. Okay, so interferon gamma will activate cytotoxic T cell and T cell and will activate the macrophages. Can you remember why?
To present more.
No?
Focus on activate killing.
Yes, macrophages are phagocytic, eat all the time, but do not digest. So after interferon gamma or in the presence of interferon gamma, the macrophages will kill the engulfed bacteria. So that's TH1. And TH17 against extracellular pathogen. So who will be the target cell? Extracellular pathogens. Who can kill the extracellulars? Bacteria, fungi, macrophages, or mostly who is the killer? Natural killer.
Neutrophils.
Neutrophils, okay. Intracellular, extracellular, very simple. Share it. Yeah. We have different cells, different mechanisms to kill. Okay. So TH17 produces IL-17, it's easy to memorize, to activate the neutrophil granulocytes. Good. One more.
Interferon gamma macrophages.
Natural killer cell cytotoxins, the intracellular. So we have a fourth group of helper T cell, follicular helper T cell, which activate the B cell. Okay? Nothing else. So the follicular helper T cell, as the name indicates, again, stay in the follicle, remain in the lymph nodes, and activate the B cell. Okay? So 4 different possibilities. Okay. So we finished the function of the helper T cell. We finished almost everything. What's the time? 6:30. So I give you 2 minutes.
2 minutes what, break?
2 minutes pause, but you cannot go out. Okay? And in 2 minutes, I would like to everyone to listen to me for 5 minutes. Okay?
Alright.
Let's bless a bit.
Are we gonna get more chairs? Are we gonna get more chairs? What are you doing?
Can you give me a list of the